Hello and welcome to our video on antigen-presenting cells, MHC Class II. The major histocompatibility complex, MHC, is a collection of genes coding for MHC molecules found on the surface of all nucleated cells of the body. In humans, the MHC genes are also referred to as human leukocyte antigen, HLA genes. Mature red blood cells, which lack a nucleus, are the only cells that do not express MHC molecules on their surface. There are two classes of MHC molecules involved in adaptive immunity, MHC1 and MHC2, figure 18.2.1. MHC1 molecules are found on all nucleated cells. They present normal self-antigens as well as abnormal or non-self pathogens to the effector T cells involved in cellular immunity. In contrast, MHC2 molecules are only found on macrophages, dendritic cells, and B cells. They present abnormal or non-self pathogen antigens for the initial activation of T cells. Both types of MHC molecules are transmembrane glycoproteins that assemble as dimers in the cytoplasmic membrane of cells, but their structures are quite different. MHC1 molecules are composed of a longer alpha protein chain coupled with a smaller beta-2 microglobulin protein, and only the alpha chain spans the cytoplasmic membrane. The alpha chain of the MHC1 molecule folds into three separate domains, alpha-1, alpha-2, and alpha-3. MHC2 molecules are composed of two protein chains, an alpha and a beta chain, that are approximately similar in length. Both chains of the MHC2 molecule possess portions that span the plasma membrane, and each chain folds into two separate domains, alpha-1 and alpha-2, and beta-1 and beta-2. In order to present abnormal or non-self antigens to T cells, MHC molecules have a cleft that serves as the antigen binding site near the top or outermost portion of the MHC1 or MHC2 dimer. For MHC1, the antigen binding cleft is formed by the alpha-1 and alpha-2 domains, whereas for MHC2, the cleft is formed by the alpha-1 and beta-1 domains, figure 18.2.1. Functions of MHC Class II. Major function of MHC Class II is to bind peptide antigen and present to CD4 T cells. CD4 plus T cells are specific for MHC2. Activates B cells for antibody production. MHC2 plays a significant role in graft versus host response and in mixed lymphocyte reaction (MLR) because the immune response gene is identical to MHC2 in humans. MHC2 are polymorphic, and various MHC2 alleles show linkage disequilibrium to a variety of autoimmune diseases. These cannot be linked entirely to the MHC2 allele, implying further involvement of genetic and/or environmental factors. For example, 95% of patients with celiac disease express an MHC2 molecule. Shown in Table 1 is a comparison of the principal differences between MHC1 and MHC2 molecules. All nucleated cells in the body have mechanisms for processing and presenting antigens in association with MHC molecules. This signals the immune system, indicating whether the cell is normal and healthy or infected with an intracellular pathogen. However, only macrophages, dendritic cells, and B cells have the ability to present antigens specifically for the purpose of activating T cells. For this reason, these types of cells are sometimes referred to as antigen-presenting cells, APCs. While all APCs play a similar role in adaptive immunity, there are some important differences to consider. Macrophages and dendritic cells are phagocytes that ingest and kill pathogens that penetrate the first-line barriers, i.e., skin and mucous membranes. B cells, on the other hand, do not function as phagocytes but play a primary role in the production and secretion of antibodies. In addition, whereas macrophages and dendritic cells recognize pathogens through non-specific receptor interactions, e.g. PAMPs, Toll-like receptors, and receptors for opsonizing complement or antibody, B cells interact with foreign pathogens, or their free antigens using antigen-specific immunoglobulin as receptors, monomeric IgD and IgM. When the immunoglobulin receptors bind to an antigen, the B cell internalizes the antigen by endocytosis before processing and presenting the antigen to T cells. The route of processing for exogenous antigens for MHC Class II presentation begins with endocytosis of the antigen. Once inside the cell, they are encased within endosomes that acidify and activate proteases to degrade the antigen. MHC Class II molecules are transported into endocytic vesicles where they bind peptide antigen and then travel to the cell surface. MHC2 molecules are only found on the surface of APCs. After a dendritic cell recognizes and attaches to a pathogen cell, the pathogen is internalized by phagocytosis and is initially contained within a phagosome. Lysosomes containing antimicrobial enzymes and chemicals fuse with the phagosome to create phagolysosome, where degradation of the pathogen for antigen processing begins. Proteases, protein degrading, are especially important in antigen processing because only protein-antigen epitopes are presented to T cells by MHC2. Figure 18.2.2. APCs do not present all possible epitopes to T cells. Only a selection of the most antigenic or immunodominant epitopes are presented. The mechanism by which epitopes are selected for processing and presentation by the APC is complicated and not well understood. However, once the most antigenic, immunodominant epitopes have been processed, they associate within the antigen-binding cleft of MHC2 molecules and are translocated to the cell surface of the dendritic cell for presentation to T cells. Let's review some key concepts and summarize. Major histocompatibility complex, MHC, is a collection of genes coding for glycoprotein molecules expressed on the surface of all nucleated cells. MHC1 molecules are expressed on all nucleated cells and are essential for presentation of normal self-antigens. Cells that become infected by intracellular pathogens can present foreign antigens on MHC1 as well, marking the infected cell for destruction. MHC2 molecules are expressed only on the surface of antigen-presenting cells, macrophages, dendritic cells, and B cells. Antigen presentation with MHC2 is essential for the activation of T cells. Antigen-presenting cells, APCs, primarily ingest pathogens by phagocytosis, destroying them in the phagolysosomes, process the protein antigens, and select the most antigenic, immunodominant epitopes with MHC2 for presentation to T cells. Thank you for watching and have a great day.